Thank you very much. Thank you very much for the kind introduction. And then it's my great honor to have the opportunity to present my work in this great symposium, great conference. Okay, so my talk is about the IntraVital microscopy technology, which can be used to do in vivo live cell imaging in a live anesthetized animal model. So my lab has been developed the IntraVital microscopy system itself, and then we try to establish the technique, the optimized technique for various tissue and organ. By doing that, we can do real-time cell level imagings. And then through many collaborations, we have done a lot of imaging analysis in various kind of human disease animal model. Okay, so this is kind of a typical data we obtain in my groups. So this is the 2-hour time-lapse 3D stack images obtained from the anesthetized mouse lymph node. So in here, what you are looking at is the red one is the T cell, green one is the B cell, and blue is the FRC, it's a fibroblastic reticular cell, which surrounds the endocellular cell inside the lymph node, actually. Okay, so you can, directly see how these two types of different lymphocytes, T cell and B cell, extravasate from the blood vessel inside the lymph node to the parenchyma of the lymph node. Okay, so this IntraVital microscopy is a very good technology if you are interested in the cell dynamics in terms of migrations and then their interaction with the different cells. Okay, so in this particular example, we use the T cell that express DS-led fluorescence protein, and B cell express GFP, green fluorescence protein, and FRC is labeled by injecting antibody, the anti-ELTI7 antibody conjugate Alexa-Fluor 647. It's far at flow 4. And then we inject this antibody conjugate directly into the peripheral tissue. And then this antibody, drained through the lymphatic vessel to the lymph node, and then it labels the FIC very specifically. And by doing that, we can we can observe these 3 types of cells simultaneously in live mouse. So this is the picture of my microscopy setup. This is a full custom build, very high speed, high power laser scanning confocal and 2-photon microscopy setup. And then this is custom built. So in my lab, we selected the individual parts. We purchased the individual parts and then assembled them like a Lego blocks, and then to make them work as a full laser scanning microscopy setup. So these are the lasers. When you can see the mirror, lens, filter, and detector. These are the laser scanner and the electronics. And then we have a stage in here. So this is an open system, actually. So we can see the laser beam coming out. We combine them into one position and then make a scanning pattern and then deliver it into the mouse tissue and then get the images. So again, this is a full custom build. So depending on the application, we can easily modify this system. So it's a very flexible system. And every component in the system is optimized for in vivo imaging of a mouse or lab models. And then this is very fast scanning system, so we can obtain about up to 100 frames per second with this image size, 512 by 512 pixels. And then both of confocal and 2-photon imaging mode, we can obtain 4 different fluorescence color imaging in simultaneous manners. And then in vivo condition, we can easily achieve sub-micrometer resolutions. So in my lab, we have developed the technique to optimize for very different kind of tissue and organs. And then, so at least in the mouse model, we can obtain the real-time cell level imaging from almost all different kind of tissue and organs, which includes like bone marrow, brain, lung, various kinds of tumor, pancreas, liver, kidney, and even heart. So this is what I do. So this is the very typical preparation for the imaging. So we first anesthetize the mouse, and then we insert the rectal probe through the anus to monitor the body temperature, and then we use the heating pad to keep the mouse temperature to be stable during the imagings. And in many cases, we catheterize the tail vein. To do that, by doing that, we can inject various kind of different drugs, fluor and even cells when we do the imagings. And then we need to expose the target tissue. So in this case, it's a lymph node, popliteal lymph node, it's here. So we make an incision on the skin and expose the popliteal lymph node, and then we put another warm water circulator to make the tissue temperature to be stable. So this is our another ongoing work in the lymph node. So inside the lymph node, we have a very specialized blood vessel, which is called high endocellular venule, HV. And then inside the HV, it has two cell layers, the endocellular cell and then FRC. So in this data, so this is the time scale we are fast forwarding, the real-time data. And in here, the green is the T cell. It's adaptively transversed. And then this red one is endocellular cell. It expresses DS-LED. And then again, this FRC is labeled by injecting this ERTL7 antibody conjugate. And then what you are looking at is T cell just flowing inside the blood vessel. And then first they attach to the, okay, let's see again. So, okay, it now attaches here. And then now it's contacting to the endocellular cell, and then they do the paracellular transendocellular migration, and then getting into this narrow channel in between the endocellular cell and the FRC. Okay, that was just a short movie. Now it's a 2-hour 3D, time-lapse movie we obtained from the lymph node. And what you're looking at is you can see, many T cells come into this HV and then do the extravasations. So this is the motions. Okay, so here, so now I will see again. So it attaches from here and then do the intravascular crawling here, and then do the transendocellular migration at this point, getting into these two cells, going up in the perivascular channel, and then do the trans-FRC migration to the parenchyma of the lymph node. So this is the first step of the multi-step, trans-endocellular and trans-FRC migration of T cell to get into the lymph node from the blood circulations. Okay, so I will show another example. So this is the preparation for the liver imaging. So again, we anesthetize the mouse. We make an incision on the skin, peritoneum, expose the liver, and then this is the example data we obtained from the liver. So in this particular example, we use type 2 GFP mouse. In this mouse, all the vascular endocellular cell express GFP. So in here, the green is the vascular, liver-sinusoid endocellular cell. And this is the real-time data, of course. And then what we did was we injected this 500 nanometer diameter porous silicon nanoparticle. It's obtained from the mauropharynx groups in University of Texas, Houston. And then we do the intravascular injection. And then, as you can see, this intravascular injected nanoparticles circulate the body and then arrive at the liver within 10 seconds. And then they quickly adhere to the endocellular cell in here and here. And then later, it's, transendocytosis to the liver parenchyma, and then later it's excreted to the small intestine later. But anyway, so this is the kind of example data we can do real-time monitoring of nanoparticle or drug carrier in the target tissues. And then the good thing of IntraVital microscopy is, during the imaging, mouse is alive, of course. The small animal model is kept alive. And then after we finish the imaging, we can recover the mouse. It means we can follow up the single mouse model in repetitive manner. So this is one example. So in this case, we make a hepatic ischemia reperfusion injury model. And this is the one mouse, one single mouse, and this is another mouse. And then what it did was in this case, we used the CX3CR1 GFP mouse model. In this mouse model, macrophage and monocyte highly express GFP. So this is right after the injury, this mouse and this mouse. And this is 24 hours after the injury. And then this is the same mouse. And then as you can see, these are the mostly Cooper cell. The shape of the Cooper cell was changed. So it means it's activated. Most of them are activated. And then you can see a lot of newly infiltrated GFP expressing cell. Most of them are bone marrow, from the bone marrow derived the monocyte, actually. So you can see this highly increased number of monocyte infiltration to the liver. And this is one drug testing data, actually. So we inject some candidate drugs, and then this candidate drug very effectively reduce this monocyte infiltration to this injured liver tissue. Then later, it gives a better outcome in the recovery process. So this is a very good example. We can use IntraVital microscopy technique to monitor the efficacy of a new drug candidate in very detailed manner in cellular level in a single mouse model. So that was just 24 hours, and then we can do this kind of observation for much longer time point, like up to 6 weeks, and then monitor the fibrosis in these cases. And then maybe some of you know that if we use two-photon microscope, we can quantitatively monitor the collagen fiber in the live tissue in vivo without any labeling. So this is the data. This is the one good example data we obtained from the NASH mouse model in the NASH and liver fibrosis model. So in here, the green is actually collagen fiber, and red is actually the lipid droplet inside the hepatocyte. So from the surface, you see the liver capsule, and then you see this very sinusoidal collagen fiber in the liver tissue. So this is a kind of a well-advanced liver fibrosis tissues. And then which obtained, of course, this data was obtained from the live anesthetic mouse livers. This is another example about the lung imagings. So in this case, we intubate the mouse, so keep the mouse to be alive, and then we expose the lung. So we make an incision on the skin, and then expose the thoracic cavity. And then since the lung is, always moving to breathe, so to get the stabilized imaging in microscopic scale, we designed this imaging chamber. So inside this imaging chamber, we have a hole here, okay? So we can suck out the air from this space through this hole to the outer pump. And by doing that, we can achieve the negative pressure inside this imaging chamber and then stabilize this part of the lung to be stabilized temporarily. And then we can observe this and then we can, get this kind of cellular level imaging from the lung of the mouse model. So this work has been done by my MD PhD student in Ba, and then work published in earlier this year. Okay, so in this case, this is a sepsis-induced acute lung injury model, and then this timeless image data was obtained at 3 hours after the modeling, the LPS high-dose LPS injections. And in this data, the green is actually blood plasma. So in green, we inject the high molecular weight hepatitis dextran, and by doing that, we do the fluorescence angiography. And the red one, this red dot is actually intravascular neutrophil. This neutrophil was labeled by injecting anti-Ly6G antibody conjugate with alloxapulvar647 again. So this is, and then by intravenously injecting this antibody conjugate, we can very effectively label the intravascular neutrophil, and then we can observe the dynamics of this neutrophil inside the pulmonary microvessels in here. As you can see, very quickly, this is only 10-minute time-lapse movie. This neutrophil make a large cluster, and then it completely block this arteriole here within just 3 or 4 minutes, like this. And then this blood flow, this upstream blood flow was completely stopped. So this is definitive data shows the neutrophil can disturb the pulmonary microcirculation in this sepsis-induced acute lung injury model by forming this cluster, intravascular cluster inside the blood vessel of the lung. So this is the sequential data, as you can see. So this cluster, neutrophil cluster formed, and then it effectively blocked the blood circulations. So this is a good kind of good representative data. Shows this we can use IntraVital microscopy to investigate the cellular level pathophysiology of various human disease model. And in drug development, we can also use this IntraVital imaging in various steps of drug development. First, from the this is the review paper published by Paul, at 2014. He's from Australia, actually. But anyway, so it's already kind of commonly accepted that we can effectively use IntraVital imaging to profile the candidate drugs in preclinical animal model at the late stage of the drug development. But as I showed in here, up to here, we can also use this IntraVital imaging for the range profiling of the target for raging research. And then maybe some of you know that we can implant various window chamber to monitor the tissue or organ for very much longer time period, much longer time window up to several weeks to several months. So in my lab, we have implement our own imaging chamber for brain, or memory tissue, and various abdominal organ or subcutaneous tissues. So this is the very well-known technique, dorsal skin fold chamber. So we can image the subcutaneous tissue for a longer time period. So in this example, we expose the subcutaneous tissue and then seal this subcutaneous tissue with the cover glass. And then in the middle, we inject the GFP-expressing cancer cells and then observe how they grow and how they respond in vivo conditions. And this is the data. This is our recent result in collaboration with Professor Yoon in Purdue University. And in this example, we make a tumor model, the breast cancer tumor model, using MDMB231 GFP cells. And then we label the blood vessel, again, injecting this anti-CD31 antibody, which label the endocellular cells. And then we inject the nanoparticle. It's shown in red here. Okay, it's better. So now we can see from 2 hours to 24 hours time frame, this intravenously injected nanoparticle arrived at this tumor model, tumor tissues. And first it appears in the perivascular area. And then later we can see the more increased delivery of this nanoparticle. And then later they deliver it into the tumor cell and then do their actions. So this is another good data. So we can use IntraVital microscopy to monitor the delivery of the drugs. Well, in this case, it's nanoparticle to the target tissue. In this case, it is cancer cell is the target. And then so we can quantitatively analyze in very high spatial temporal resolutions. And then this is another observation to monitor the blood vessel in vivo in every 3 days. Okay, well, these days we started to look at the immune cell interaction with the cancer cells. So this is just one data. Okay, due to the time limitation, I maybe should go fast. So this is another data in memory imaging window. So in this case, we can image the memory pattern. And then this is So this is the dissemination of the cancer cell into the blood circulation observed by using those memory imaging windows. So you can see this GFP expressing cancer cell now get into the blood vessel and then go into the systemic circulations. And then this is the cranial window model. So by implanting this cranial window, we can observe the cell level changes over 10 days after inducing the neuroinflammations. And then later at 3 weeks, so this is the same site actually, up to 1 month. And then now we can see at 3 weeks after this 3MP injections, we can see the breakdown of the BBB, and then later it results in the establishment of the huge inflammatory response in these particular areas. So again, these temporal changes in the same tissue in the live animal model can be effectively investigated by using IntraVital microscopy techniques. This is the glioblastoma mouse model. And then change of the blood vessel, which happens very fast. Only after 48 hours, you can see the huge dilation of the blood vessel and the remodelings. And then this is the data we obtained from the pancreas cancer model. And then this pancreas, spleen, also this is kidney. So we can, basically we can image almost every tissue and organ in vivo conditions. And the one limitation about this IntraVital microscopy technique using confocal and two-photon microscopy is the limited imaging depth. Even with the two-photon microscope, the maximum imaging penetration depth is about 500 micrometer, or up to 700 micrometer into the tissues. So this is the kind of a fundamental limitation we are now facing. So one way to overcome this limitation is using this very narrow diameter lens, which is called green lens. It has a cylindrical rod shapes with a very small diameter from 0.35 to 1 millimeter. And then we can, assemble this lens like this. And then we can use it as like a laparoscope or a side view endoscope. So in this example, we inserted this endoscope through the lip bones and then access the heart. And this is our result obtained from the heart of the live mouse model by using this microendoscope. And then recently we inserted this endoscope into the needle like this, and then we can insert this needle into the solid tissue, in this case it's brain, and then make it as a side view. So while we're inserting this side view endoscope, we just record the imaging data, and then now we can have this depth-wise visualization of the in cellular level in solid tissues. So now we kind of overcome the imaging penetration depth limitation. Although it's a little bit invasive, but the diameter is like 0.7 millimeter now. So it's minimally invasive, let's say. And this is another data we obtained from the brain. And then we can do the same approach in the tumor models, up to several millimeter, like more than 5 millimeter. And then still, we can observe that we can obtain the cellular images. Okay, so maybe I should go faster. So in case of a tooth model, we recently established the clearing technique for the mouse tooth. And by doing that, we can do the 3D visualization of the full mouse tooth. Although it's not in vivo, it's ex vivo conditions, but still, we effectively cleared the hard tissue of the dentin and enamels. And then by doing that, we can observe the internal cellular level distribution of the cells inside the mouse tooth in the dental pulp. Okay, maybe I spent too much time. So, okay, this is the conclusion. So, IntraVital microscopy can enable a real-time imaging of various cellular level dynamics, as well as such as cell trafficking, cell migration, cell interaction, and cell microenvironment interaction inside the living body in vivo. And then, it can provide various new insight in the process of human disease development and treatment. And for the drug development, this IntraVital microscopy can enable a direct analysis of delivery to the target tissue or cell, and their subsequent efficacy, and we can also validate the mode of action of a new therapeutic candidate in much detailed manner in microscopic cellular level in various preclinical models of human disease. So maybe I should finish my talk here. Thank you very much.